Good morning, sir.
Good morning, Namrata.
Good morning, sir.
Good morning, good morning, Manaswini. Namrata, Manaswini, call your all things to join.
Okay, sir.
मेरे एंड में ना they already created their team MSc हो चुका है INC PhD फर्स्ट सेमेस्टर but it is the thing is it's a very वो सेशन टाइम वो नहीं हुआ तो लगे तो है ये ना ऊपर से इनका जो भी they know people they do whatever they we always have to तो मतलब कराने कॉम्पिटिशन आगे भी आता है
Okay, students. So now most of you are here, so we can start. Okay. So these slides are visible to you. The slides
Yes.
are visible to you.
Yes, sir. Yes.
Okay, so one more thing. So all of you from today onward, so please try to join on time because this joining time is going to, you're taking quite long. So many are joining very late and so even some of you are joining at the end of the class. So those who are joining at the end of class will be marked absent. So be sure and make yourself sure that you should join within the 5 minutes of start of the class. Like if your class is starting at 12:15, so you must be in the team by 12:20. Okay. If anyone is going to come later than that, then I will consider them as an absent. Okay. Okay, so we are starting with what we have to do today. So as in our previous classes, we have done some of the introduction things, and I will also told you about what the course I'm going to teach you. And the course name, as you all are aware about, and you all have chosen your APW. So I think you most of are aware about it, and you all, I think you all have read your syllabus also. And the title of the course is The Fundamental of Recombinant DNA Technology. So simple, if you remember that in our previous class, we talked about what this recombinant means. And we are doing something with the DNA and some techniques are going to be there. So all these three terminologies which combine together and that leads to the our current subject. And the module 1 that we are going to deal with, and the module that we are going to start with is the module number 1, that means is the enzymes. So what are different enzymes which are being involved in your recombinant DNA technology, that we are going to deal with it in this module. So just I'm naming the enzymes that we are going to study with the restriction endonuclease, ligase, polymerase, reverse transcriptase, kinase, phosphatase, nuclease, tyrosine deoxytransferase, topoisomerase, linkers, and adapters. So these are the contents of your module number 1. Okay, so now moving to the thing that we are talking about. So before going to the details about the enzymes, so first ever when you are going to do anything, it's our duty to remember the historical events or the researchers whose discoveries and whose research made us to come at the current level. So whenever you simply, if you are reading any book, it simply means thousands of the scientists have done their work, which are being compiled into those books. In the financing, simply when you say books, in that books you around at least 10,000 of researchers are going to be involved in the book, that the content that book have. However, the authors, you will find that in the books there are one or two authors, or maximum 10, 20 authors are there in your books. But uh, around 10,000 researchers are there behind the discovery, or behind the research, or behind the content that we are dealing with in that book are going to be there. Okay, so where the some timeline is there, and the timeline describes some important discoveries. So the first, if you see, in 1869, and the scientist named Leisure, and the important role of him is that he isolated the DNA, because the name that we are talking about, recombinant DNA technology. So the first thing, because if you don't having the DNA, if you don't know what DNA is, if you are unaware about the DNA, then you can't do anything. So the first discovery was 1869, because without the DNA, earlier than this, you were not aware about that. What is genetic material? You might have in your genetics also the genetic material which carries the information from generations. So here the first in 1869, we came to know that this measure he told you that the DNA, he isolated the DNA. Then came to 1944, so a large gap is there, because the discovery of DNA came in 1869. But you are not aware about that, what for what purpose the DNA is. So that came into 1944, that every provides the evidence. He gave the first evidence that it is the DNA which is your genetic material, which carries the genetic information. Because earlier we believed that protein is the one who is behind the genetic information, because the protein is the one which carries the function. It is true, but today also the protein carries the function. But when you are talking about the genetic things, which carries the information, it is your DNA, and this information came into the first. You can simply say that around uh, 75-year gap is there between the discovery of DNA as a to the discovery of DNA. Okay, so then after around 10 years in 1959, Watson, Crick, and Wilkins, these four are the pioneer researchers who decipher the structure of DNA that you all know today. Is you're talking about the Watson-Crick model of DNA. Watson-Crick structure of your DNA was deciphered by these four researchers: Watson-Crick and Franklin and the Wilkins. So these four together they generated the information, they generated the data, and on the data generated by Franklin and Wilkins, Watson resolved the structure, and that you talking about the dual helical structures, or the DNA helix that came into the 1950s. So once you know that this is the DNA, then you can do the genetic material, and Watson discovered that this is the structure of DNA. Now these key discoveries, they speed up the research, and he found that earlier time you find that a 100-year gap, around 75-year gap, 10-year gap was there. In 57 years, there were 4 year, Kornberg he discovered the DNA polymerase. So polymerase is the enzyme which are behind the replication. And now in case of your recombinant DNA technology, polymerase have a great role in that also. And then again after gap of 4 years, in 1961, Marvin and Doty, they discovered the process of DNA, the denaturation. So that the process in which your DNA that you are talking about, the DNA is double helical, two helix are there. They can get separated, and after the separation, they can come together also. Means this is the
Excuse
renaturation,
me, sir.
denaturation things are there.
Sorry
Again,
to
that we are knowing about.
interrupt you in
Yes.
between. Sir, your voice is breaking a lot.
My voice is like to all or only to Namrita?
So your voice was breaking.
Yes, Namrita, it
Sir,
was not breaking.
it was breaking a little, but now it's better.
Yes, sir.
Okay, now its voice is fine.
It's slightly better, sir.
Is my because
Yes, sir,
I'm at the
there's
campus,
a
so
lot of
there should
background
not be
noise.
background noise also. Okay. Okay, so I can't do anything in that because I'm using the university net, and I hope it is quite fast also. So the network issue might be at your end, which is because of that my voice is cracking at your end. And if it is breaking to all, that definitely is going to be problem at my end. But at the moment, I can't do anything with that. Okay, so is voice better now? Is my voice better and audible to all?
Yes, sir.
Yes,
Okay.
sir. It's audible.
Yes, sir.
Yes. Okay, so please all of you turn off your mic also, so that will also reduce the background noise. Okay, so we were talking about the history and what was the contribution of different researchers. So we were at 1961 that we talked about the discovery of that thing that the denaturation means your DNA that you are talking about that is double stranded. You can make it to the single stranded, and you again the double you can again renature it to the single double stranded form. So this also become the basis of many of the techniques that we are interested in your further in your course further, like PCR that you are talking about, PCR is there, and hybridization that you talk about, the southern hybridization, the one northern hybridization thing is there. So all these things, the basis of those, all the thing that you are talking about, that's the basis of the Marmar and Doty's discovery. Then for the 1 year gap, Aber he provided the first evidence that DNA restriction nucleases are there. That restriction nuclease simply means that there are the enzymes are there which can break the DNA. So the Arbor deciphered that we can break the DNA. And this discovery of these enzymes, that the restriction nuclease is which are able to break down your DNA. That's become the basis of for that nowadays you're talking about sequence are there. So in your another microbiology class, you might have heard about how to define your DNA. So DNA is simple way you can define your DNA. It is a sequence of, or it is a polymer of nucleotides. Okay. The nucleotides you also describe in terms of bases, but just calling the bases doesn't describe your DNA. But if you call the nucleotides, then it describes you completely. And that the sequence of nucleotide is your DNA, or a polymer of nucleotide is your DNA. But when you're referring to the base also, that also not a wrong, because two nucleotides that we are referring, that is like your adenine, guanine, thymine, cytosine, all those four nucleotides that we are they differ by their bases. Your sugar and phosphate backbone is common. All the nucleotides they are going to have sugar and phosphate, but the difference is there is your nitrogenous base. So adenosine or adenine, that different it is the adenosine is your nitrogenous base. Similarly, in case of adenine is nitrogenous base, guanine in case of and so on. In case of thymine and the side of all the differences. Sugar is in case of your DNA, your deoxynucleotide, it is deoxyribose sugar. RNA, it is ribose sugar. And the phosphate is also there. things. So both in case of your RNA and DNA, so there is no difference on the basis of your sugar and the phosphate. So difference between two nucleotides is there only on the basis of your nitrogenous base. Okay. So then come to the 1966. Now the main thing discovered when Indian is involved in it, so that's why we are putting more emphasis on it. This is the Nuremberg, Otero, and the Corona. So what they did, they elucidate the genetic code. So genetic code means the information that are there in your DNA that is codified in the form of a triplet of your three bases are there. They are together. They are coding for one information that is called the genetic code. One code code for amino acid. So this is the described by the Nuremberg method and Corona. So then come to the next year, that is the 1967. Now the further more few enzymes were being discovered that were done by the Gellert. So what he did, he discovered an enzyme that called ligase. So the job of the ligase is joining. So another thing when you're talking about the recombinant DNA technology. So here you can utilize this enzyme, you can join two nucleotides together or two strands together or two fragments together. In detail we will further study. Okay, then coming to the 72, 73. So now further more things are coming up. Boyer, Cohen, and Berg, what they did, they decipher, they describe a new technique, that is the cloning technique. Cloning, you all might be aware about. Clone means two identical things. So generation of the identical things, we are calling the clones. So if you generate two identical human, we are calling them a clone. But in case of your recombinant DNA technology, we generate DNA clone, means two same nucleotide sequence strands we are generating, or two same sequence we are generating, so that we are the clone. So how we are going to generate it? This technique was described by the Boyer, Cohen, and Berg in 1973, and then comes to 75. Now the next thing came, that is we are talking about southern blotting. So means the DNA that you are present in your gel. So some techniques we were studying in the laboratory also. So you can transfer your DNA from to the membrane. So membrane means you can immobilize your DNA. So the immobilization process or the transfer process we described over the southern. So then the name of that we will call the southern blotting. Then the further more discoveries came, the Sanger, Maxim Gilbert, and they got the Nobel Prizes also Sanger. So what he did, he deciphered the technique to find the DNA sequence. So because how to get the DNA sequence, what the DNA sequence is. So other discoveries we are talking about, they are telling you these are the techniques you can know the code, but we want the sequence. So earlier the sequence techniques were on the basis of degradation, means keep on degrading your nucleotide and keep on finding what nucleotide is coming out. But now some more advanced techniques came into the picture that come with the discovery of the Sanger and sequencing that we are talking about these days. So most of the sequencing are done based on the basis of Sanger sequencing. And at the same time, Maxim and Gilbert also describe you the sequencing things, but most commonly we are using Sanger sequencing because that is more easier than the Maxim Gilbert sequencing. So then further in 81 and 82, more things came, and now the recombinant technology established itself, and the process of development of transgenic games means not just you are generating the recombinant DNA, but you are also transferring to an individual, transferring to an organism, so that you can generate a transgenic organism in case of animal and in case of plant also. So these are the bit more discovery keep on going on. Then the Nobel discovery came in 1955. That is that you all might be knowing the name Carey Mullis. And what he discovered, he did his work co-worker, he discovered the PCR, which is simply called polymerase chain reaction. So nowadays, in today's time, your recombinant DNA technology and whatever you say, your molecular biology can't survive with the discovery. That discovery is done by the Carey Mullis. So as nowadays you are all talking about, in case of the COVID pandemic, you are all at home, and the diagnosis of your COVID is done by the RT-PCR that you are talking about. So that PCR technique is based upon the discovery of the Carey Mullis. So without the work of the Carey Mullis, without acknowledging the Carey Mullis, we will not be able to diagnose your COVID. Okay. simple word you can say that this 1985 discovered this. It is a breakthrough discovery of PCR, so which break all the, and it's also advanced too much that in every type of every type of your technology, your molecular biology technology, or your life science technology, we are utilizing the PCR. So wherever you name it, PCR is going to be involved. Okay, so no techniques is going to be there in biology where the PCR is not going to be involved. By this or that way, PCR is going to be involved. Because wherever the gene is going to come, you are going to talk about the PCR. Okay, so this is just a brief history, and what help, these are the few researchers which helped us in the establishment of the current concept of the recombinant DNA technology, how we are going to form further. As I mentioned, that recombinant DNA technology simply means the combining DNA. When you are combining DNA, when you are generating a new DNA, there are going to be involved tools. As without tools, you can't do any engineering. So here also in our DNA, recombinant DNA technology, in our technology, we require some tools. So in case of who is a software engineer, that's going to use some software, some tools, electronic tools. He's going to be a mechanical engineer. He's going to have mechanical tools, and vehicle transport engineer. He's going to do that type of tools are going to be there. And we are as a molecular biology things. We are as a genetic engineering. We are as a DNA engineer. We need our type of tool. So in our case, the tool that we are using, first tool is your enzymes. So since you are 11th, 12th, I think you are talking about, you are studying about the enzyme. So enzymes, the role of enzyme is what they do, they catalyze the reaction. What type of the reaction they are going to catalyze, you name them. So means there will be going to be a substrate, and the enzyme will convert into the a product. Okay, so the conversion of substrate to product is the job of enzyme. Here also our enzyme also going to do the similar task. So what task we want to do in case of our recombinant DNA technology? We all, as I'm talking about since our previous class, that we are going to play with DNA. So how to play with DNA? What type of things we need to do with the DNA? So DNA, as we are talking about, DNA is a segment or a fragment or a chain of nucleotides that you're talking about. So what we can do, we can cut it, we can extend it, we can join it, we can make copy of it, we can modify it. Modify means we are not breaking it, we are not adding up more nucleotide to it, just we are adding some more groups to it. Simply, or you know that would be, I talked about the phosphate thing is there. So you want to remove the phosphate or you want to add the phosphate. That is the modifying thing. You want to add a hydroxyl group. You want to remove a hydroxyl group. As you know that ribose is there and deoxyribose are there. So DNA, you want to convert DNA to RNA. You want to convert RNA to DNA. So how to do that? So for that, the enzymes that you need a modifying enzyme. You can modify it. So you can convert it to DNA to RNA. You can also convert to the RNA to DNA. So these are the different types of enzymes. So basically, we classify, we group our enzymes that we are going to utilize in biology, basically in the 4 groups. So these are the 4 groups. One is the nucleases. So as the name there, nuclease. So means nuclease is something that your nucleic acid is going to be involved, and nuclease means the activity that they are going to do. They are going to provide a breakdown activity or the degradation activity or a shortening activity. Means a long stretch of DNA is there, they are going to break it. And when they are breaking it, so once the activity is done, you are going to have 2 fragments. Okay, so their job is the breaking. So like simply if you see that like this is a pen, if you are breaking, so this is the job. Earlier it is this one, now 2 things are there, so 2 fragments are there, so simply you can say. So this is the job of your nuclease. It break down your DNA, it cut your DNA. So how it is going to cut, what type of cut is going to produce, that we'll study in the future slides. But just the first type of enzyme that you are talking about nuclease, its main role is breaking down, cutting it. Right? If my finger is there, if you want to cut it, then you want to remove this finger, and then you need a nuclease if it is made up of DNA. So similar, this is the job of your nuclease. Next type of a class of enzyme that you are going to study are there's a molecular recombinant technology that we need for recombinant technology, your ligases. The ligase means joining. So the job of ligase is the joining of nucleic acids together. Simply means you are having two fragments of DNA, like first one nuclease break and make two. So ligase bring two fragments together and make it one. This is the job of your ligase. Okay. So this is the second class of your enzymes. What type of the ligase are there? How you are getting that? How they are doing? What their process is? We will discuss in detail later. And the next type of the enzyme that you're talking about, like next class, is the polymerase. So polymerase, the name polymer is there. As the name polymer is there, the job of this enzyme is the polymerization. So simply polymerization means you are going to have a small, what polymerization is required in case of your DNA. So DNA are the polymer of your nucleotides. The job of polymerase is bring down nucleotides together and making a long chain of your DNA. So this is the simple job of your polymerase. So polymerase means if you give the nucleotide to your polymerase, it will form a DNA. DNA, a sequence of your DNA. So how it is going to do, what is the requirement? So it can either create a de novo strand, or it can't create a de novo strand. That is the requirement of your polymerase, which we will discuss in detail later. And the next class of the enzyme, as I mentioned, the modifying enzyme. So modifying enzyme is the one which is modifying. Modifying means it is going to add some group, some additional group you want to add. Like simple, if you want to add a methyl group, so methyl means CH3. So if you want to add a CH3 over your DNA, so if you want to add sulfur over your DNA, if you want to add a phosphate to your DNA, okay, or if you want to add some to your DNA simply, if you want or any group of came in your mind, you say methyl group you want to add, not just the methyl. So if you want to add any type of the group to your DNA molecule and you want to study, if you add a methyl group to it, so what type of function it will change. So that for that another class of enzyme that you need to modify enzyme. So these enzymes, this is the fourth class. We will discuss in detail in future slides. So as you can see, so you are having further more type of enzyme. The kinases are there, so they are going to come into modifying enzyme, and phosphatase is there. This is also modifying enzyme, and terminal deoxytransferase is also modifying enzyme. This topoisomerase is also modifying enzyme. So rushing endonuclease, it is type of nuclease is ligase, so polymerase and reverse transcriptase both are the class of your polymerases. Okay, so now coming to the first type of your enzyme, so that is the nuclease or nucleases. So as I mentioned to you when I was talking about the introduction brief about the previous slide, so nuclease means it is going to degrade. Degrade means, as you can see in the okay, so as you can see, this is each you can say this is a nucleotide. So these all two nucleotides joined by the sugar phosphate backbone, and they are joined together. So nuclease, as mentioned, it is job of the cleavage. So cleavage simply means what it's going to do. It is going to break the phosphodiester bond. So two nucleotides, they are joined together by phosphodiester bond. So this phosphodiester, the job of this nuclease is the breaking down the phosphodiester bond. So now the simple question came. So for everywhere, every strand, every nucleotide, two nucleotides joined together by a phosphodiester bond, and two strands of DNA, they are joined together by a hydrogen bond. So these two strands, they are together by the hydrogen bond, and two nucleotides joined together by a phosphodiester bond. So in both the strands, the strand number 1, whose double stranded DNA is there, two strands you are showing. So both the strands are joined by the hydrogen bond, and in the strand that is supposed to have a nucleotide sequence, they are joined together by a phosphodiester. So what these nuclease enzymes are going to do? These nucleotides, they are degrading DNA molecule by breaking a phosphodiester bond that links one nucleotide to the next in DNA strand. So now simple jobs came. So which nucleotide, which phosphodiester bond your enzyme will break? So you can say that every nucleotide has the phosphodiester bond. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So 17 phosphodiester bonds are there in your DNA strand, you can say. So now you simply may ask where in your DNA it is going to produce a cut. So where it to produce, so here the term breaking this phosphodiester bond, we are utilizing term a cut. So when you say uh, now we are not going to talk about, we are not going to say that it is breaking the phosphodiester bond, but we use a term cut instead of a phosphodiester bond. But if anybody asks you what nucleotides do, you have to mention that it is going to break a phosphodiester bond. Okay. Then in running form, you can say, you can call the cut. It produces cut, where it is going to produce cut. So simply if you say, so now you can see that the way to produce cut, which phosphodiester bond it to break. So now on the basis of where it is going to produce cut or which phosphodiester bond it is going to break, we are divided into two types. So your nucleases are of two types on the basis where they are going to produce cut. So where they are producing cut means whether they are producing cut within or at the ends. So here you can see that the two types of things, but these are the ends. This is one end, this is second end. Okay. So if your nucleus is going to produce cut at the ends, so if it is doing cleavage at the end, then we are calling it as an exonuclease. So what is going to occur? If it is producing cut at the end, then individual nucleotides being released. Okay. So if it is producing cut here, so these nucleotides, you can see these nucleotides are going to come out. So if it is producing cut here, so two nucleotides are going to be out. Okay. So then after producing one cut, now this one more end is going to be generated, and it again produce cut. So simply means if you keep these nucleases, those nucleases which are producing cut at the end, so then we call the exonuclease. The end product that you are going to have is going to be the nucleotides, because as the end is going to be generated, it will produce a new cut. So that at the end, what you are going to have, you are going to have a nucleotides. Okay, simply means it is removing nucleotides from the ends. One. So those nucleases which are removing nucleotide from the ends or which break the phosphodiester bond at the end, or those which produce cleavage at the end, or which remove nucleotides from the terminus, we call the exonuclease. So here the term exo from your schooling or your 11th 12th, you are using term exo for the outside. So here it also can use the term the outside means the ends. So nucleus which produce cut at the ends, you call them the exonuclease. Is it clear? Is it clear?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So nucleases which are producing cut at the end, we call them the exonuclease. Okay. So now a second type which produce cut within, which do the cleavage within. So within means endo. So the thing that cleavage, if it is happening within, then you call the endo. So if the cut is being produced here, so simply you can say that this cleavage, it is, if the cut is produced at this point. So what are you going to generate? You are going to generate two fragments. So here you are not going to have uh, nucleotides, but you are going to have a fragment of DNA. So then again this your endo means nucleus is there, it can further can produce the cut within, so then it will again produce the cut, so you again will get the fragment. So simply means you can say that these nucleotides are not going to produce cut at the ends, but they are producing cuts within your DNA strand. So such type of the nucleus we call the endonuclease. So simply you can say that the exonuclease remove the nucleotides at one nucleotide one at a time from the ends. So कितने remove करेंगे? They are going to remove one nucleotide at a time. That is going to be from the ends. But in case of the endonuclease, what are going to do? They are going to break internal phosphodiester bonds. So बीच का अंदर का inside the phosphodiester bond that is there, they are going to produce cut. and when they are going to produce cut there, that leads to the generation of the fragments. So fragments of DNA are going to get. Whereas in case of the exonuclease, we are going to get nucleotides, free nucleotides plus only one fragment. Okay. So this is the thing. So now coming to the in detail about the exonuclease. So what is exonuclease are there? What type of the exonuclease? So here also further you can classify them. So now you may ask that you are having double stranded. So in case of your double stranded, you all know that the DNA have the two strands. One strand run from the 5' to 3'. Second also run in the 5' to 3', but in the reverse direction. So which end they can produce cut. So a nucleus may be there which can produce cut at the both the 5' and the 3' end, one chance, or a nucleus which are producing cut only at the 5 terminus or only cut at the 3 terminus. So on this basis, we further classify your DNA you at which end they are going to produce cut. So the difference among different exonuclease lies in their number of strands they are degrading. So number of strands they are degrading means both the strands they are degrading or single strand they are degrading. Those who are degrading double strand, we call them the double stranded cutter, means they are cutting your nucleotides, they are removing your nucleotides from the both the strand of your DNA. Simply means, so here you can see the first case. Okay, so here you can see when it is the cut at the both ends. So this is the 5', this is going to the 3', this is going to the 3', and this is going to be a 5'. So it is breaking at the 3' and the 5'. So both the strand they are cutting, then we are going to have a double stranded cutter. So the example of this as enzyme name is the BAL1, BAL 31. So the name is enzyme BAL31. So the how this BAL31 came? This came from the B strand came from the bacterium, A came from the AL came from the Altromonas. So this is the BAL1 and 31 is the number of your enzyme. So which number enzyme is being isolated. So the from the bacterium Altromonas ST Giana, the enzyme that you isolated, which is the exonuclease in nature, name of that enzyme is the BAL31. So BAL31, what is doing? It is producing a double stranded cut, means it is removing the nucleotide from the ends from the both the strand. Okay, but a second type, that is a single stranded cutter. So single stranded cutter is also depending on which end it is producing cut, whether it is producing cut at the 3 terminus or it is producing at the 5 terminus. So here the example that is given here, the name of the enzyme is exonuclease 3. So this exonuclease 3 is enzyme isolated from the bacterium again. The name of the bacteria is E. coli, Escherichia coli. So from the Escherichia coli enzyme isolated called exonuclease 3, what it is doing? It is producing single strand cut and it is removing nucleotide only from 3' terminus. Okay, so here you can say this is a 5', this is a 3', so 2 3' are there. So this enzyme, if you add to this DNA, it will produce cut here and it will also produce cut here and so on. Okay, so what you are going to add in the end? So when this it keep on cutting it, so the point came when the both the enzyme will join and they will remove the single strand. So single stranded DNA will get at the end plus the nucleotide. So these are the endonuclease. So two types of endonuclease that we mentioned. One, they are producing cut on the both the strands. One which is producing cut on a single strand. They are not producing cut on the double strand. Means they are not breaking both the 5 phosphodiester bond of your 2 strand of your DNA. Okay, is it clear?
Yes, sir.
So I have one doubt.
Yes.
So the for exonuclease 3, it removes from only the 3' terminus, but it's both ends, right?
Yes.
As shown in the okay.
Both that. So
Thank you.
wherever it found the 3', it will remove. Okay, if it is equivalent 3' OH and it will remove. It won't remove from the 5'.
Okay, sir. Thank you.
Okay. Okay. So now this is the one type. Now another type of the exonuclease is also there, that is called the endonuclease. So the term endonuclease means when it is producing cut within. Now endonuclease are of the depending as we previously described what type of endonuclease are, whether they are cutting single strand, they are cutting double stranded DNA. As we talk about their endonuclease, they are producing cut within. So here are some example of the enzymes are there. Now you may ask yourself where within it cut. So within there are also from the two nucleotides. So and you're calling that this is the like just a moment. So so if it is producing cut from here, then you call it the exonuclease. But if it is cut from here, then you are calling it as a endonuclease. So this is a simple difference between the endo and exo. You can say if it is a terminus being removed, we are calling it an exonuclease. If within the terminus it is being removed, then you are calling it the endonuclease. Okay, so this one is exo, this one is endo. So now you may ask one question. So sir, where within? Does it produce cut here? It will cut here, it will cut here, that we will talk about later in this. So, but for the one thing that we are discussing here, the types of endonuclease. So, endonuclease, as you described earlier, two types of exonucleases are there. One, they are cutting a single strand. Second, they are cutting double strand. So, here also two types are there. One you call that that is from the that you are got, getting that the name of the enzyme is your SI nuclease. So what it do? Endonuclease from Aspergillus coryzi you got it. Only cleaves a single stranded DNA. Deoxyribonuclease 1 is prepared from a cow pancreas. What it do? It cut from the both the double stranded DNA. Now the two types of enzyme you are having, one is your SI nuclease and second is your DNA is 1. Okay. So DNA is 1. If you have the if you take the your SI nuclease, if you give the double-stranded DNA to it, it won't produce any cut. It won't cut it. It will cut only if you give a single-stranded DNA. Okay? If you give a double-stranded DNA to your SI nuclease, it won't produce any cut. Okay? So now, second thing that we call the your SI sorry, or your SI nuclease. So sorry, one thing I told one thing. So one thing, in case of your SI nuclease, what it's doing, the upper one is going to be SI nuclease. So it is if you are producing single strand DNA, it is producing cut and provide the fragments. So as I mentioned, this 2 is the one which is going to be the endo. So at the end, the point what you are going to have, you are going to have a 2 nucleotides in a way. Okay, depending on where it is going to produce cut. So in this case, you may get the 3 also. So this is the 3 one. But this 3 one, if you produce cut here, then the job of an exonuclease will come. So your endonuclease won't produce cut at this point. So if you either you are having at 3 or 2, so the fragments which are going to be generated after the activity of your endonuclease is going to be your either 2 nucleotide fragment or a 3 nucleotide fragment. And now the second thing is your if you provide a double stranded DNA to it. So what it's going to do in that case? Now in this case, first it will produce a single stranded cut. Single stranded DNA is there, like this is double stranded one. In this double stranded one, this fragment is there, which is single stranded. Okay. So what job of SI nuclease, it will produce only to the single stranded DNA. It is not going to produce to the double stranded DNA. So this portion is a single stranded portion. So it will produce cut here. It will produce cut here. It will produce cut here. Okay. So when it produces cut in this way, so it will remove the nucleotides and you will have the double stranded. So once these double-stranded fragment forms, now SI nuclease won't produce further cuts. So if you give the single-stranded DNA to your SI nuclease, it will produce cut. And if you give a double-stranded DNA which is having a portion which is single-stranded, then it will produce cut to there. simple term you can say, if the single-stranded DNA is available or a hanging DNA is available which is having a single-stranded component, then your SI nuclease will cut, otherwise your SI nuclease will not. Okay, now come to the DNase 1. So DNase 1, it produce a cut in a non-specific manner. So when we are saying non-specific means it will produce cut to the both, whether you are having a single stranded or you are going to have a double stranded. So if you add an endonuclease, which is a DNase 1 in nature, it will chop off every type of DNA, whether it's going to be single stranded or it's going to be a double stranded. So this will be a single stranded DNA. So we are going to have fragments, so 3 fragments, and so on. Okay. So here, in case of double stranded, so it will produce cut within and so on. Okay. So this is the way how your endonuclease are going to work. So two types, as I mentioned. One is the SI1, which produced a single stranded, and DNase1, which produced degrade both single stranded and the double stranded. Now another type of the endonuclease is also there. As in the first these two types, we mentioned that there is no specificity where they are going to produce cut. We don't know. We just know that it will two nucleotides inside they will produce the protein. Because from two nucleotides inside, we start calling it as a endonuclease. Simple. Now your DNA is very long. So if you found that any specificity is there, if you want to know that any specific site is there, any specific location is there within your DNA where your restricted your endonuclease are going to produce a cut, another class of your nucleases came. That to them we call them the restriction endonuclease. Okay, so now on the basis of where these enzymes are producing cut, they are producing cut at a single stranded DNA, double stranded DNA, or double stranded DNA at a specific site. If the DNA which are cleaving at a specific site, at a specific location, to them we call them the restriction endonuclease. Because here the term restriction is there. Because why you are utilizing term restriction here? Because you are restricting your endonuclease to produce cut only at a restricted location. If the restricted location is there, then only they will produce cut. If restricted location is not there, they won't produce any type of the cut. Okay. So any question till now? Any question till now?
No, sir.
Okay, so this is the one that endonuclease, exonuclease thing is there. So three types are there in case of endonuclease. One is the SI1, which is producing single-stranded cut. Second is the both single and double-stranded. Third one we are talking about a specific location that we call the restriction endonuclease. So simply, if you see that in the lower one, so it is going to produce at if this location is there, that will produce cut and it will stop working. It won't work anymore. Okay, so. Now, further, we are talking about endonuclease on the basis of location, as I'm talking about. Two things are there, whether they are producing specifically or the non-specifically. So, in previous slide, these two were the non-specifically way, and the lower one C1, that is the specifically way. So, the more important to us, if you want to do a specific job, then you need a specific enzyme. So when you're talking about tools, what type of tools are there? So now you know that in case of at your house, mistri and this masons are there. So there one having a hathoda, big hathoda, and those mistri was there. He was having a smaller one. And that smaller one going to do a specific job, but a bigger one they can break anything. So when you want to break a specific thing, when you want a brick, when you want to break your specific brick, and you want to make your brick size so that it can fit a space wherever you want to putting it. So for that you need a smaller one which can break, not crush the brick, but make brick in a smaller way. So you can have a specific type of the enzyme. So specific type of your endonuclease. So simply you can say that two types: one is the specific one, second is the non-specific one. So specific one is the enzyme which break your strand DNA internally, internal portion, but in specific way. So there you are calling them the restriction enzyme because their action is the restricted. So they are cleaving phosphodiester bond within the each strand. In each strand means both the strand they are going to produce the cut. Okay. So where what the specific things are there, what the sites they are restricted. So we are calling these enzymes a new name that we are calling the restriction endonucleases or the RE. So what these restriction endonucleases are there? As I mentioned, they are of very much importance, and so we are going to study these enzymes in details also, in further details, how they are working, how they have been discovered. So I'm halting today's lecture at this moment only, and in our next class we'll talk about these in details. So now few minutes are there, so we have to do with our attendance. So I will call your name, and when I call your name, recite and turn on your camera. So Adish. Adish,
Present, sir.
Adish, turn on
Present,
your
sir.
camera also. Atoiba,
Present,
Atoiba,
sir.
Ayushi,
Present, sir.
just a moment. Okay, Demetrius, Demetrius,
Present, sir.
Dev, Dev, Dev absent. Manaswini,
Present, sir.
Manaswini,
So my camera is just switching on. I'm present.
Yuvraj, Yuvraj,
Present, sir.
Sanjana, Sanjana, Nehal, Nehal,
Present, sir. Present, sir.
Athoiba, Athoiba.
Present, sir.
Why are you taking so long time, Athoiba? Devanshi, Devanshi,
Sir, Devanshi is having network.
Nandini, 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 absent. Kshitij, Kshitij,
Sir.
Kshitij, Janvi,
Present, sir.
Shivani, Shivani, absent. Shakti,
Present,
Shakti,
sir.
Rupal,
Present, sir.
कौन सा बोला है? रूपल बोला, शक्ति बोला है?
Sir, Rupal. Present, sir. Shakti.
रूपल,
Yes, sir. Present.
साहिबा, साहिबा,
Present, sir. Present, sir.
इशिता,
Present, sir.
नम्रता,
Present, sir.
तन्वी,
Sir.
तन्वी,
Present, sir.
turn on your camera also, Tanvi. Yukta, Yukta,
Present.
Ayushi,
Present, sir.
Yukta, turn on your camera. Yukta, turn on your camera.
Yes.
Okay. Vivek, Vivek?
Present, sir. Present, sir.
Okay, so absentees are Tridev is absent, Devanshi absent.
Sir, I'm present, sir. Sir, Tridev is present.
त्रिदेव भाई सोओगे तो कैसे काम चलेगा?
No, sir. I just went to the hospital, so I was just coming back.
तो
So
हॉस्पिटल गए थे, क्लास में
I
नहीं थे ना?
no, sir. I was there in the class. I was all time listening to you, and
Then you are
I
not
also
facing any
said
time of
present,
attendance?
sir. Sir, I also said present, but I think so because of the network issue, you were not able to hear me. But I was always there.
Devanshi,
Sir, Devanshi is having network issues.
Nandini and Shivani. Nandini Raj, Shivani Mishra.
Sir, I am present. Sir, I am present.
So Shivani, why you are not speaking at the time when you are not?
Sir, I said present. I said present.
From next time onward, I will not call your name second time. So be vigilant when your attendance is there. Okay, so only one absent is there, that is Nandini.
Sir, so I was present, right?
Yes, you are all are present except Nandini.
Okay, thank you, sir.
Okay, so now we are having a few more minutes. So if you want to ask anything regarding today's lecture, you can ask. So any doubts or anything that you want to need clarification?
Sir, can you give an example of a non-specific endonuclease?
So these are the example here on the DNA is one and
Okay.
nuclease
Okay.
example,
Okay. Thank you, sir.
non-specific endonucleases. Any other thing?
No, sir, not from mine.
So except Manaswini, no one is vigilant, I think. So none of you are asking. Okay, so if you no query, then you may leave the session.
Sir, Nandini ने text किया.
Yeah, I will check. Okay, so thank you all. So if you those who don't have query, they may leave and they
Thank
can
you, sir.
join the next session.
Thank you, sir.
Thank you, sir.
Thank you, sir.